Dr. Leavitt is a full professor in the Department of Medical Genetics and the Department of Medicine, Division of Neurology Associate at the University of British Columbia. Dr. Leavitt completed his medical degree at McGill, medical internship at Columbia Presbyterian, neurology residency at Cornell and Harvard. While in Boston, he completed a basic neuroscience research fellowship at Harvard Medical School and Children's Hospital of Boston. Blair is a consulting neurologist and director of research at the UBC Center for Huntington's Disease. A scientist and a physician, Dr. Leavitt's time, both clinical and research, is dedicated to developing new treatments for genetic brain disorders such as Huntington's disease. He also works on other neurodegenerative diseases, including amyotrophic lateral sclerosis and frontotemporal dementia. Dr. Leavitt is currently the director of CMMT Transgenic Animal Facility, the co-chair of the Huntington Study Group, and a founding editor-in-chief of the Journal of Huntington's Disease. And now I'd like to read to you about Dr. Lauren Boak, who I had a wonderful opportunity to spend time with just recently. Lauren has spent her career in clinical research and development. She began over a decade ago as a clinical researcher in Australia before joining Roche in Switzerland in 2007. At Roche, Lauren worked on study teams for different compounds in disease areas including infectious diseases, immunology, and neuroscience. Over the past 8 years, she has held roles on both the clinical and business side of the organization, specializing in neuroscience. Currently, Lauren is the global development lead for the Roche investigational drug in Huntington's disease. As the cross-functional development team leader, she leads a team of scientific and development experts responsible for design and execution of the clinical trial plan, HTT-ASO. Lauren is the mother of 2 young children, 2 and 5, and when not working on this incredible project, enjoys spending time with them and their dad. Though as parents, they are a little worried about keeping up with their kids' secret spoken language, Swiss German. It is with pleasure that I welcome Dr. Leavitt and Dr. Boak to the stage.
Well, thank you, Robin. Unfortunately for all of you, you have to put up with me before you get to the lovely Lauren. I'd like to thank the HSC for the invitation and the chance to speak again. It's been a while since I've had a chance to talk to you all, and it's great to see so many old friends in the audience. I think the last time was Winnipeg, so great choice, Kelowna over Winnipeg. Love Winnipeg, don't get me wrong, but I like BC as well. So I'm going to be talking today about Huntingtin-Lowering therapies, and specifically I'm going to be talking about the Roche Ionis HTTRX RG6042 project and where we're going. I'm going to lay the introduction. I'm going to give you a little bit of the background of how this came to the clinical development, and then I'm going to talk about the study that's been recently been completed. And then when it comes to the future, I'm going to pass that on to Lauren, and she's going to tell you about the future. So questions about the upcoming trials can be directed to Lauren. All right. I am going to talk about some science, so bear with me. I'm going to try and make sure we're all on the same page. There'll be lots of time for questions. If I do a poor job of explaining things, Jeff Carroll will certainly tell me how bad a job I do, but don't be afraid to ask any questions at any point, and certainly we'll have time at the end. So really important, we're going to start with some of the basics, and we're going to start with what we sort of call the central dogma of molecular biology, this basic idea that we have this stuff called DNA, the roadmap of what makes us who we are. So genes are made up of DNA. Genes are transcribed into RNA, and that RNA is translated into protein, and the protein are the building blocks of our cells. And this is going to be really important to understand this pathway, because we're going to talk about interfering with this pathway and how we interfere in this pathway to prevent a bad protein, an abnormal protein, a protein that causes disease, specifically Huntington's disease, from being produced. And that's what we're talking about today. We're talking about Huntingtin-Lowering therapies. So, probably everyone in the audience knows, but again, I want to make sure we're all on the same page. The gene that causes Huntington's disease is present in all of us. It's called the Huntingtin gene, and we all have 2 copies of it. If you've inherited the gene change that causes Huntington's disease, there's an expansion, a repeated element of repeated CAG, CAG, CAG within that gene. That is translated into a protein called the Huntingtin protein. And what we call that the mutant Huntingtin protein. I'm going to say that a lot because all of what we're doing with the therapy is trying to get rid of that mutant Huntingtin protein. There's lots of different ways that mutant Huntingtin protein can affect cells, and especially brain cells, because that's the cell that's most affected in HD. But in the end, it doesn't really matter, because what we're going to try and do is we're going to try and prevent production of that protein completely. So it doesn't really matter what the downstream pathways are if you can prevent the production of the RNA and the protein. So if you remember, gene makes RNA makes protein. The hypothesis for Huntingtin-Lowering therapy is really simple. If we can prevent the production of that mutant RNA and that mutant protein, we should be able to alleviate the pathogenic process, the disease-causing process that leads to Huntington's disease. All right, how are we going to do that? We're going to use something called antisense technology. And you're going to hear a lot about antisense oligonucleotides, or ASOs. And I'm going to explain what an ASO is. And basically, an oligonucleotide is just a scientific word for an artificial stretch of DNA, or a stretch of nucleotides. We're talking about specifically nucleotides that are about 20 to 21 nucleotides long. And by antisense, we mean that they're opposite of a strand that we're targeting. So you'd have a sense strand, that's the normal strand that you want to target, and then you'd have an antisense strand, the oligo, that will bind to it. So in this case, we have some RNA in the cell. The antisense oligonucleotide will bind to that RNA. Well, what happens when a piece of DNA binds to a piece of RNA? It forms a RNA-DNA hybrid. That's what we would technically call it. And that's not normal. That sometimes happens during viral infections or things like that, and it shouldn't be there. So the cell actually has a way of dealing with it. And in the case of the ASOs that we're talking about, there's an enzyme in the body called RNase H1 that will actually break down those DNA-RNA hybrids. So when you break down the DNA and the RNA, you don't make the protein. So that's how this particular ASO works. It binds to the Huntingtin RNA, prevents it from being translated into the protein. No RNA, no protein. So, how would ASO therapy work for HD? We have the Huntingtin gene. It makes Huntingtin RNA. Now we develop, or Ionis develop, 21 nucleotides of artificial DNA that bind only to that RNA. And 21 nucleotides is enough to be specific for every single RNA in the body. It's really incredible, but the genomic code is so complex that even a short stretch can be specific for a single strand of RNA. So we're targeting just the Huntingtin RNA. And hundreds, over 300 different ASOs have been screened to find ASOs that are selective and specific just for this RNA. The ASO binds to the Huntingtin RNA and prevents production of mutant Huntingtin protein. That's the basic principle behind the ASO that we're talking about. Why ASOs? Why not some other approaches you may have heard about? There's some real advantages to this therapy. This has already been in people. There are approved therapies based on ASOs, and they've been approved since 1998. The very first ASO was approved for use in the eye. They actually get into cells. They're diffusible. If you can get it to a cell, it will be taken up. So if you have brain cells in addition, you put an ASO on it, they'll take it up and it will work. It doesn't need a virus to get it into cells. It doesn't need anything. It just needs to get to those cells. The more you give, the more effect it has. It's dose dependent, just like what we think about as drugs. When you take a higher dose, you get more of an effect. That's really important. It's also relatively stable. So we can give a dose and have an effect that lasts for months in some cases. That's great. You don't have to take it every day. And most importantly, I think, for safety, these drugs are reversible. That relatively long-term effect isn't permanent. So if there are any safety concerns, You can stop administering the drug, and the levels of the protein come back to normal. Really important. Again, some of the other approaches that are in development don't have these safety features. So there are a lot of really great reasons why this moved very rapidly, and by very rapidly, I mean really incredibly slowly for those of us who've been following it. This was more than a 10-year development, and I'm going to tell you about some of the development. So there was a lot of work that got us to the place we are today, which is a really amazing place to be for those of us that have been in the field for a long time. So what does it take? Great idea. What does it take to get a drug like this into people? It takes about 12 years of hard work. You have to prove that you can design a drug like this that does what you say. It has to selectively bind to the RNA, it has to lower the amount of Huntingtin, and you want it to do that in a whole bunch of models. Not just one, not just two, as many different models of Huntington's disease as you can. The more data you can generate, the better. You have to make sure that it's safe, so you have to do all sorts of studies of toxicology. And I'm actually going to take you through some of this stuff. One of the big questions is, and it's come up a few times, do you have to target just the abnormal gene, or can you target Huntingtin in general? So there's such a thing as total Huntingtin lowering and allele-specific lowering, where you may only lower the mutants. And there's a very complicated math that goes into which was used, but in this case, we're moving forward with total Huntingtin lowering. And so the question was asked, is this viable, and is it beneficial to lower total Huntingtin? And the question has been answered quite clearly in preclinical models. And then importantly, I said, if you can get it to brain cells, they take it up freely. Well, how do we get it to brain cells? Well, the way we do it in this study is to put it into the spinal fluid. And when the question had to be answered, if you put it in spinal fluid, does it get to the parts of the brain it has to, and does it work? And I'm going to show you all the answers. And there is going to be a bit of science here, and I apologize, but hey, you guys should see the data, you should see it, and you should judge it for yourself. We've all looked at it. This is dose dependency. So this shows as you increase the amount of the ASO in the bottom, Huntingtin levels drop. So more of the ASO, the more the levels of Huntingtin drop in the brain of a mouse that has the Huntingtin gene, the mutant Huntingtin gene. In fact, the human Huntingtin gene. That's great. So it's dose dependent. This just shows more ASO, less Huntingtin, another graph. So very clear dose dependency. That's really important. Does it have an effect in animal models? So if you can infuse it into the brain of a mouse, you get an effect. This is actually measuring the amount of that RNA, and yes, it's down, and it's down after a single injection for as long as 4 months. So it has an effect, the effect is prolonged, but it's reversible. It does come back to normal. This is what's called the Western blot, and the density of those black bands is how much protein is in that brain. And you can see that after 2 weeks, 4 weeks, 6 weeks, 8 weeks, it's significantly lower. So not only does it affect the RNA, it affects the protein. Does it improve the mice? So these mice that have mutant Huntingtin in them have abnormalities. One of the abnormalities is their ability to run on a running wheel, a standardized test we call the rotor rod. The open squares is a normal mouse, can run on that wheel pretty well, stays on it for over 200 seconds. A mouse that has the Huntingtin gene can't do it as well. That's the black box. When you treat these mice with the ASO, you get a significant improvement. Not complete rescue, but a pretty good, and importantly, a prolonged benefit. So, it lowers RNA, it lowers protein, and it improves the phenotype. It improves the Huntingtin symptoms in the mouse. What about this question of total Huntingtin lowering versus allele-specific lowering? So, to give Ionis credit, they asked this question right up front. They had developed both allele-specific and total Huntingtin lowering approaches. And they tested total Huntingtin lowering versus allele-specific, and both of them had similar levels of improvement. Total Huntingtin lowering approaches treat everybody. It's a single drug for everyone. It's the first one that went forward. This is just some pretty pictures. It shows in red little dots, that's the actual ASO labeled with fluorescence. The green on the top is staining for brain cells, and you can show that the red spots or in the green brain cells shows that the ASO gets taken up into brain cells. It also gets taken up to some of the other cells in the brain. Lynn Raymond was talking today about the astrocytes and microglia, and those cells take it up too. So not only does it get into neurons, it gets into the supporting players in the brain, the astrocytes and the microglia. And this is just some pretty pictures that show that happened for the other science geeks in the room. So this is a picture of the non-human primate brain. And this is pretty similar on a small scale to the human brain. So what happens if you administer this now to a non-human primate, a monkey, in the exact same way we want to do it in people? We're giving it into the spinal fluid. Does it have an effect on the Huntington levels in the brain? And sure enough, in the regions of the brain affected by Huntington's disease, in the cortex, the caudate, the thalamus, it lowers both the RNA and the protein, as predicted. So it works in small animals, works in big animals, and it's been tested in a whole variety of animal models. So there's a huge amount of preclinical data to show that if you administer this drug, if you put it in the spinal fluid, it gets to the brain, and it lowers both the RNA and the protein that cause this disease. And it's prolonged. In this case, even out to 8 weeks after a single infusion, there were significant lowering of Huntingtin, both in the deep gray structures of the brain, the caudate putamen, striatum, and the superficial part of the brain, the cortex. It's greater in the cortex, about twice as much in the cortex as the deep gray structure, but it lowers them in both. So I'm not going to show you all the toxicity data other than to tell you that extensive work has been done in a number of animal models, and this had a very favorable safety, even at doses many times higher than were ever used in people. And again, in non-human primates, you can put this in the brain and follow them for many, many months, up to 15 months, and even longer now, without showing any abnormalities. So it looks like it was safe. So, going back, can an ASO be developed that selectively lowers Huntingtin levels, lowers RNA, lowers protein, and improves the Huntington symptoms in animals? Yes. Is it safe in all of these tests of toxicity? Yes. If you give it into the spinal fluid, does it get to the brain and does it affect the regions that it needs to? Yes. And if you target total Huntington versus just the alleles, just the mutant allele, is that still beneficial? The evidence is yes. So I've said a few times that we're putting it into the spinal fluid. How do we do that? I think most people have probably heard of the spinal tap. Basically, you put a needle between the bones and the lumbar vertebrae into what's called the intrathecal space, the space that surrounds the spinal cord. That CSF is continuous with the CSF, the cerebral spinal fluid, that the brain floats in. Essentially, there's a column of fluid in our spinal, a column and our brain float in that fluid. And if you put a freely diffusible drug like an ASO, even down here, if a person gets up and walks around, within 30 minutes, that ASO is distributed throughout the entire CSF volume. And that CSF stays there for a long time. It slowly exchanges over about 8 hours. So you slowly make it and you slowly produce it. But basically, it's like a column of fluid. And you put the drug in, it rapidly distributes. So, before the study started, it was realized that what would be really helpful is if we could measure Huntingtin in the brain. We want to give people this drug and we want to show that it's lowering Huntingtin in the brain primarily. That's really hard to do in people. We could do brain biopsies, it's possible, but nobody wants to be having bits of their brain taken out. So we were really lucky. We had been working with cerebral spinal fluid for a long time, and just as this trial was ready to go, two groups had identified a way of measuring mutant Huntingtin in the spinal fluid. One of whom was led by, I think everyone in the room's hero, Ed Wild, including mine, he's one of my heroes too, and others, to develop a really sensitive way of measuring mutant Huntingtin in the cerebral spinal fluid. And this was done with samples from our group and samples from the London group, and shown that in people that don't have the mutation, you don't measure any mutant Huntingtin, and that's not a surprise. In pre-manifest individuals who don't have the disease but carry the gene, you can measure it, and as they get closer to disease, the level gets a little higher. And in individuals who have a diagnosis, the levels are high and measurable. That's great, but is there any evidence that the mutant huntingtin in the brain, actually, in the spinal fluid, actually reflects the levels in the brain? That's what we really want. I mean, there are other ways to do this, and there are some things in development, imaging techniques, that have been mentioned in a couple of other sessions where we might be able to use, say, a special PET ligand and do PET imaging and measure the amount of huntingtin in the brain, but those aren't ready yet. Fortunately, this was ready, and we asked the question, well, can we prove that the mutant huntingtin in spinal fluid is actually coming from brain? And a group led by Michael Hayden and Amber Southwell in his lab came up with a couple of really cool experiments. And I think this is the last mouse data I'm going to show you, and then I'm going to stick with people. But again, using a slightly different approach, they looked at the CSF from our group at UBC, and again showed that as disease progresses, the levels of mutant huntingtin go up. And importantly, they did some really neat experiments in mice. So using a genetic trick, they created mice that had mutant huntingtin everywhere except the parts of the brain we really care about, the cortex and the forebrain. And those mice up there are called back DXP mice. And if you see, there's that little band of mutant huntingtin that's missing. It's missing in the forebrain. So we created mice that have huntingtin everywhere except the forebrain, and then when you look at the CSF, that's that black bar there, it's actually dramatically decreased. So pretty good evidence that maybe huntingtin is coming from the forebrain. If you don't express it in the forebrain, you don't get it in the spinal fluid. Well, why is it going up as disease progresses? The belief is that injured cells release this. So what if you actually go ahead and injure cells? You do something that Lynn Raymond has talked a lot about, and you increase the excitation in the striata. You give a toxin that kills brain cells. When you do that, you get a spike 3 days after you do it. That's that little spike there. Okay, so it looks like if you don't have huntingtin in your brain, you don't get it in your spinal fluid. If you have injury, you get more. Well, what happens if you take a mouse that has mutant huntingtin in their brain and you give them an ASO? So we did that. If you give them an ASO, you decrease the amount of the protein in the brain, and it goes down in spinal fluid. So there's what we call a correlation. If you look at brain huntingtin on the bottom and CSF huntingtin, the more you have in your brain, the more you have in your CSF. So this is the data to suggest that mutant huntingtin in the spinal fluid is what we call a biomarker. It reflects the amount in brain. And that's going to be really important when we talk about the human clinical trial, which is what I'm going to talk about next. So good evidence that cerebral spinal fluid mutant huntingtin reflects brain. So now I'd like to tell you the results of the first Huntingtin-Lowering trial, the Ionis HTTRX agent that's now known as RG6042. It's the total Huntingtin-lowering ASO developed by Ionis and carried forward now by Roche. As I mentioned, this was a huge amount of work. This started in 2005, and the first patient didn't receive an infusion until 2015, so a full 10 years of very careful development. This doesn't happen overnight, and it's a huge collaborative effort, both by industry and by academics, to make this happen. This is an ASO that targets total huntingtin. It's administered by an intrathecal bolus push, which basically, as I said, means you do the equivalent of a spinal tap. You take off some fluid, you put back some fluid with the drug in it. And we know that that distributes to the brain very quickly. This is the first time this has been done in people. It's what we call a first-in-human or first-in-man trial. The very first thing we need to show is that this is safe and tolerated. Can this be done? And that's really the primary outcome measure for this study. A successful study would be to say we can do this, and it's safe, and it's tolerated, end of story. We want to look at a few other things, obviously. We want to see whether or not it has an effect on mutant huntingtin in the spinal fluid. We want to see how the drug distributes in the brain, all of those things in plasma and CSF. And then at the end, if we can see any other effects, we want to look for all of those as well. But this was first and foremost a study to show is it safe and is it tolerated. So I'm just going to tell you a little bit about how the study was done. This is what we call a placebo-controlled double-blind study. There was a randomization, so for every 3 people that got active drug, 1 person got placebo. It's double-blind. Neither the investigators or the individual participants knew what they were getting until the end of the study. And then there were 4 successive doses, 28 days apart. And then a follow-up lumbar puncture after that. I'm going to show you in a little bit more detail how that was done, just so you understand some of the subsequent results. So the very first dose, which we call day 1, was actually preceded by a removal of the spinal fluid. And every time we do a dose, we take off the same amount of fluid as we give back with the drug. So when I tell you about the spinal fluid, we're actually looking at the spinal fluid in what's called a trough. What trough means is it's the furthest point from the last dose. So we dose at day 1, at 28 days, just before we give the next dose, we take spinal fluid, and that's what we're going to be looking at. And we did that each time, and then we took a final spinal fluid at some point after the last dose. Participants were well matched. The other thing that was really important about this study is not only were we looking at safety, but we wanted to get some information about how different doses work. I've shown you that there's a whole decade of preclinical animal information that told us how these doses affect animals. We don't do it in people. So we have to start at a very, very low dose and then gradually bring that up in the safest way possible to ensure we're not doing any harm. So we started with a tiny little dose of 10 milligrams, and we waited until we had enough data from that first group, and there was a separate group of investigators who looked at all the data to make sure there was no harm coming to the patients, that everything was safe. And then once we had enough information, we were able to go up to the next highest dose. And we had to follow them until we knew that was safe. And then the next highest dose. Oops, sorry. And that continued gradually until we were able to get up to the maximum dose that we treated. And again, at each of these dosing intervals, some people got placebo and some people got the drug. So there are some people that got a very low dose and some people that got a high dose. And we were able to look at the effects of those different doses. So, what were the results of the study? The single and the most important result was that this was safe and well tolerated. There were really no serious and significant adverse events. There was only 1 hospitalization. It was a placebo-treated individual who stayed overnight because they had a bit of a headache, was fine the next morning, no treatment initiated. All 46 of the individuals in this study worldwide completed the study, are currently being treated in what we call an open label, where we know they're getting active drug. And all 46 individuals have continued to get treated in the open label study. So we have excellent data to suggest this is safe. There are this procedure does come with some potential risks and side effects. It's not uncommon to have a small amount of pain in the lumbar region afterwards. There is a needle going in there. Anyone who's had a blood draw knows sometimes you get a bit of pain where a needle goes through the skin. Headaches do occur, but there have been no headaches so significant they required any specific therapy to date. So, safe and well tolerated. The study was a success. I could stop there and pass it on to Lauren, but there's a little bit more information I think you probably want to hear about. How about Huntingtin Levels in the spinal fluid? I wouldn't have told you all that preclinical work and all of our beautiful CSF work if there wasn't a reason to talk about it. This is what happens to mutant Huntingtin levels in the spinal fluid at the different doses. The gray dots are placebo, and they basically average out around no change. As you increase the dose, you get more and more significant decreased levels of mutant huntingtin in the spinal fluid. It might be best to look at that over time. So this is the 2 highest doses, 90 milligrams and 120 milligrams on the left. And you can see that with each successive dose, you get lower and lower levels of huntingtin in the spinal fluid. This is a dream result. I have to tell you as a scientist, I wouldn't have expected it to look this beautiful, but it does. It's the data. You gotta one thing scientists love is good data. This is great data. So, you know, I mentioned there was a huge amount of preclinical work. And we actually knew from that preclinical work where we wanted to get to. We wanted to get to levels of CSF around somewhere between 40 and 60%. We don't necessarily want to eliminate Huntington, especially not since this is total Huntington that we're knocking down. But if we can get it down to 40 to 60%, we know what that should reflect in brain levels. So in the cortex, the outside of the brain, there's actually a little bit greater than what the CSF shows. So it's probably somewhere between 50 and 85% knockdown. And in the deep gray structures, the caudate, putamen, and thalamus, it's probably somewhere between 20 and 45. And that's really important. That's the target. We hit that target, and that's the range in animal models that showed benefit. So we believe we're giving a drug that's safe. We have a measure in the brain that we think reflects brain levels. We're hitting that target, and it's a target that is probably going to be effective. We don't know, but it's effective in all of our preclinical models. So that's promising. Did we see anyone improve? Everyone asks. The simple question is yes, everyone improved. Everyone always improves in studies. That's why we have to do placebo-controlled trials. But this was a very short study. It was only from start to finish 7 months. And there's not a lot of change in that brief period of time. And if you look at any single dose and any single measure, there were no significant changes. End of story. But this was a successful trial. It was safe. It was well tolerated. And we saw a significant effect on mutant Huntingtin levels. Well, we can look a little closer at the data, and this is purely exploratory. This does not mean we have any evidence that the drug works or not. This is just an exploratory analysis. But we can look and we can say, okay, well, we know some people's Huntingtin levels went down. How did they do on a bunch of the clinical scales? So now we start looking at everyone all together. And I'm just going to skip ahead to this graph here. If we look at everyone and we graph how much did Huntingtin levels change from the start of the study to the end of the study, and we look at a number of different measures. This is one that combines motor function, cognitive function, and total functional capacity. So it's a composite scale. So it sort of looks at progression in general. And this is a scale that changes by about 1 point in a year on average. And there's some variability, obviously. But if you look at the correlation, that line shows the average line through these points. The more Huntingtin was lowered, the better people tended to do on this scale. So it suggests, and correlation does not mean causation, it does not mean that this drug works, but if you look at it, the people that had the most Huntingtin lowering did better on this scale and did better on a number of scales. So that again is is just suggestive, it's not evidence or proof, but it's what we'd hope to see. You hope that if we lower Huntingtin, we see an improvement. Less Huntingtin, better scores on this. Again, it's not proof, it's just suggestive. So, what did we learn? Drug is well tolerated, safe at all doses tested. There was a dose-dependent reduction in mutant Huntingtin. We also know that it's reversible because after we treated patients, the level did return to normal. There were no individual group, single doses didn't change any individual parameters, but in exploratory analyses, the more Huntingtin was lowered, the better there were on certain scores. And I'm not going to tell you about the subsequent studies, that's up to Lauren, but I do want to say a few things. This is just, again, a brief summary of the results. I'm not going to say again, but I think I've made my point. Just want to thank a few people. This was done in 9 sites in Europe and our site in North America. That's a picture of Sarah Tabrizi, for those of you who don't know her. She's a dynamo. She's a wonderful friend and colleague, and really was one of the driving forces. There were a huge group of individuals, both at Ionis, CHDI, Roche, and of course our staff at the clinic. But most importantly, There were 46 individuals who participated in a first-in-human trial. They're the real heroes. They're the ones who deserve any credit for this, or not any credit. They're the ones who deserve the most credit for this. They're the ones who committed themselves in the time. So I have a couple more things to say. But yeah, thank you. That's important. I just actually want to say thank you to the HSC and to Brain Canada. Intrathecal administration is possible. There are now individuals who had have had 18 of these infusions or lumbar punctures. So it can be done. We've done it safely now. But we all would like to have easier means of administration. The Huntington Society and Brain Canada have seen fit to reward a group of investigators from across Canada, including our group at UBC, actually led from our group at UBC, to develop new ways and better ways of getting these ASOs across the blood-brain barrier. So thank you to the Huntington Society of Canada and Brain Canada. And also, the Huntington Society of Canada has also funded a clinical fellowship at UBC. And this is the Manning family and the Beckman family clinical research fellowship. So training the next generation of physicians to do these things, because God knows I'm getting older by the day. So thank you again to the Huntington Society of Canada, the Manning family, and the Beckman family for the research fellow. Now I'll say thank you. I think we'll go through our presentations and then open it up for questions at the end.
Hello everyone. I think there's a bit of furniture removal.
Yes, exactly.
Shall I get started? Next neurologist and okay, so thank you for your time. I'm so excited and privileged to be here. The Huntington community is truly, truly unique. I've been working on the Roche program for just over a year, and it's really, really incredible getting to know all of you in the community. I want to thank Robin and HSC for the opportunity to give an update on the program today. As mentioned, I'm Australian, and so I have a fond appreciation for the Canadians. I have got a lot of Canadian friends in Basel, actually, where I currently live. I actually live in Switzerland. I've been there for almost 12 years. My badge actually says I'm working in the Mississauga Roche office, but that's not the case. And it's just a truly incredible experience to be working on this program. And so I'm so happy I can be here today to give you an update. I'm on behalf of a very big team at Roche. As mentioned, I'm a development team leader of this program, and what that means is I am leading a group of experts. There's truly a village, an army being put together to move this program forward, and it's truly a very exciting and promising time, and I hope to be able to share some information that you're seeking today. And yes, so I'm very happy to be here. Firstly, just because I work for a pharmaceutical company, I need to share just some disclosures at the beginning. I'm just going to read this from the screen. This presentation contains general information about our Huntington's program and is not intended as specific medical advice. RG6042 is an investigational, that means not approved, medicine that is being studied for the treatment of people with Huntington's disease. RG6042 has not been approved by health authorities, including Health Canada or the FDA in the US. The effectiveness and safety of RG6042 are currently being studied. So you should talk with your healthcare provider for information and advice about your condition, including any current or potential treatments. Okay, so now moving on to my presentation. I'm going to start just with a very quick introduction to Roche. You may not know much about Roche, and so I wanted just to start with that. Also, just a bit of the history of the program. Blair has taken you through this in great expert detail, but just also touch on some of the aspects that Roche has been involved with up until this point. And then clearly the part of the presentation you're no doubt keen to hear most is about the update of the clinical development program and the next steps in clinical trials. And then we'll have Q&A at the end. So firstly, Roche. So the overall mission of Roche is to transform the lives of people with serious illnesses, such as Huntington's disease. I'm not sure if everyone knows, but Roche started in Canada in 1931. It was a small facility outside of Montreal, I believe. But now we have over 1,000 employees across both a diagnostics division, also the pharmaceuticals division. So there's a small site outside of Montreal, and then a larger site in Mississauga, Ontario. And in particular, in Mississauga, there's teams working on clinical development and clinical trials for many of our programs at Roche, including in oncology or for cancer. And now we're building that team for the Huntington's program as well. Just a few stats. Roche has been around for a very long time and is truly, to its mission, has a number of medicines, 30 actually, on the WHO Essential Medicines List. And this is a medicines list that, as the name suggests, is essential for health across the world. I really love working at Roche. It's a truly science and driven company. And just last year, in Canadian dollars, $52 million was invested into research. Importantly, there are a number of Roche products already on the market that already treat Canadians, and the stats for this is 315,000 patients received a Roche product last year. But now to this program. And it truly is an exciting time for the Huntington's disease community. We've been very patient, and there's still a lot of answers to be found, but it truly is marking a important time in research and development for Huntington's. And it's not only Roche, but our colleagues in the field who are moving programs forward. Just want to take a step back quickly, just about how Roche has been involved with this program. We didn't just come onto the program at the end of last year. We actually have forged a partnership with Ionis over 5 years ago. And so when Roche was involved together with Ionis to decide which molecule would be taken forward into further testing, then that led to the start of the phase 1/2A study that Blair outlined in September 2015. In parallel, Roche has been collaborating with the community and HD specialists, including Blair and Ed Wild, on a number of activities. Importantly, this CSF mutant Huntingtin assay. And this has become a true pillar for this program now, with the exciting data to show that this molecule can reduce mutant Huntingtin in the CSF. Other activities we've been closely involved with, but collaborating with the community very closely, is around endpoint development. So if we do have a drug to test in humans to see if it benefits HD patients, how are we going to measure that benefit? And so we've been working on a number of efforts with the experts in the field on clinical and digital endpoints. And these are coming into the future studies. Then at the end of last year, as you all know, the phase 1/2A study read out. And with this exciting data and the collaboration with the community, Roche decided to license this investigational medicine from Ionis. And so this is when the time point where Ionis HTT-RX turned into RG6042. And don't ask me what the 6042 means, but RG, R stands for Roche, and our US subsidiary Genentech is the G. Now, from this year onwards, this is when we've been really gearing up activities at Roche for this program, and to initiate the registrational programs that I will go into more detail in a moment. But another really important activity that we've been doing since we've sort of come out from behind and taken a more active role in this program is deepening the relationships with you and the community. And I just want to spend a moment just talking through Roche's philosophy on the community and partnership with the community. So we are responsible for the development of this molecule now, taking it to the next phase of clinical testing, and hopefully, if all the promise pays out, an effective treatment that patients can access. But we're not doing this for the HD community, we're doing this with the HD community. And so that's why it's a real pleasure to be at your meeting today. But I will also highlight some of the other activities we're doing with the community. The first is through HD Cope. Now this is an initiative that HSC, together with HDSA from the US, have put together, and EHE, the European Patient Association. And this is what we were involved with at the start of the year is an advisory board, a patient and family advisory board consisting of HD Cope members. And this was a real unique experience for us to hear it from individuals from HD families, gene positive carriers or caregivers, what their expectations are for the next phase of development of this molecule, in designing the study, in considering the burden of clinical trials. We've learned a lot from this HD Cope group. The other aspect I would like to highlight is that we have what we are terming the Strategic Advisory Committee. And this consists of HD specialists, including Blair, from across the world to help advise us on our program and what we need to do, and just work together to build the best program and the best clinical trial protocols to test the effectiveness of this drug, this investigational medicine. But also on this Strategic Advisory Committee, we have two community members. We have a caregiver, as well, two caregivers and one gene-positive carrier. And so this really helps us make sure that we're considering the concerns, the wishes, and the perspectives of HD families and caregivers as we move this program forward. Now this slide, you've seen a version of this from Blair. And again, I just want to reiterate, and I think also we've seen it nice, beautifully drawn in the session with Jeff and Tam earlier. What we are doing with this particular molecule, RG6042, is reducing total mutant and total protein, so both the mutant Huntingtin protein and the normal or wild type protein. And this is through ASO, which is basically marking the RNA for destruction and leading to no production of the mutant Huntingtin. Just want to just go into a little bit more detail of some of the thought process and design process that went in at the beginning of this program. Blair's already outlined this, but I just want to reiterate a couple of aspects also. When Ionis came to decide which molecule they would move forward into clinical testing, one of the real considerations was whether or not this molecule has the potential to treat all HD patients. And so this was a key decision that was made together with Roche to go down the total Huntingtin-Lowering path versus the allele-specific path. Because at the time, we had data, the preclinical data on the potency and safety, so how we expect the drug to work available for the two different approaches. There was no distinguishing this, and so this was the important attribute of the total protein Huntington approach that was used. Again, I don't want to take too much time again, but this ASO leads to partial reduction of protein, where I have the ability to titrate or adjust the dose if we see a safety issue, and by nature is reversible. So we do, that allows the flexibility to stop dosing or halt dosing if there is a problem. At the same time, we also need to have regular dosing, or how frequent yet we don't know, to maintain effect. So far, the safety and tolerability in animals and humans is looking good for this molecule, and it's this confidence and this wealth of data that Ionis has built together with the field over the years that has given us the confidence to move this program forward for the HD community. Just building on that again, so when we start thinking about what clinical trials we need to do next and how we're going to move this molecule to the next phase, we have a number of questions that we need to answer still. The exciting phase 1/2A data that Blair outlined was such a great start for this. But now what will happen if we treat for longer, or when we treat for longer than the 13 weeks in this phase 1/2A study? Clinical benefit. Now this is really what we're looking to see. We've lowered mutant Huntingtin. The science suggests that this is going to be beneficial, but we need to prove this. And this is what the next clinical studies will do. The key question is, does sustained treatment slow or stop the progression in Huntington's disease? Equally as important is whether or not this drug provides benefit, it's what the safety is over a longer period of time. So the patients in the initial study received 4 doses. They are now in an open label extension study, and some patients have received up to 10 doses of study drug. This, we would expect to have to treat for a much longer period of time with this. So the key question for the next round of studies is, what will the long-term safety look like? And the last question just to raise here is around dosing. Monthly intrathecal administration or lumbar punctures of an ASO is quite a daunting proposition. If it's needed, then that's what's needed. If monthly is needed to lead to the benefit that you are looking for, then that's what we'll need to do. But we do want to explore whether or not we can dose less frequently than monthly. So these are another consideration that we had in mind when designing our program. So, now, what is next? These brave 46 patients in the phase 1/2A study, as mentioned, have all rolled over into what we call an open label extension study. And in this study, all patients are receiving active drug. There are 2 arms in this ongoing study, so we're already starting to test the differences between a monthly regimen of RG6042 and a less frequent dosing regimen of every other month of RG6042. So there's 2 what we call arms in this study. 23 patients are receiving monthly 120 milligrams of RG6042, and 23 patients are receiving a dose every other month. So what we call bi-monthly. This is ongoing. and all patients were enrolled by the middle of this year. Now I want to take you through the 2 next studies that we are planning and will be starting by the end of this year. The first is what we are calling the HD Natural History Study. This is an observational non-drug study. And Generation HD1, which is the phase 3 pivotal study. The HD community has collected a lot of data, a lot of observational data over the years. What now is in the Enroll platform, over 16,000 patients within this. So there's a wealth of data that already exists on the natural history study. On the natural history, or the changes over time in the clinical symptoms and outcomes in individuals with HD. But there are still many unanswered questions. And this HD natural history study aims to answer some of these. In particular, looking at the role of mutant Huntingtin protein in disease progression. So this study was looking at the relationship between changes in mutant Huntingtin over time with changes in clinical status or disease progression in HD. This study will be starting by the end of this year. It will be conducted in Canada, US, UK and Germany. Up to 100 patients with stage 1 or 2 HD will be enrolled. And the study will last 15 months in duration. There'll be a baseline visit, a visit at 3 months, a visit at 9 months, and a visit at 15 months. So this study already has more visits than the traditional observational study that are being done, are currently ongoing in HD. So HD Clarity, which is a sub-study of Enroll, is a study that some of you may be aware of, where CSF samples are collected every year. Actually, even 2 years. Maybe it's annual and maybe it's at least annual. No more frequent than annual. There's also a UCL CSF study that Edward is leading, and I know that that's the samples are collected every 2 years. So this will provide more. Oops, I've lost my pointer. Okay. So it's going to provide a richer data set than currently exists. And this is going to be really important, going to answer some critical questions about the role of mutant Huntingtin in disease progression. Another important point just to raise is that clearly there's a commitment and a burden to participate in an observational study like this, that there's no hope for benefit. So provided that data in the program still supports treatment, these patients that enroll in this study will be offered open label treatment at the end of the 15 months. Okay, so that's the natural history study. I'm going to be starting imminently, so we're very excited about this. Now I'd like to move on to the phase 3 study. What we are calling the global evaluation of efficacy and safety of Roche Genentech antisense oligonucleotide for Huntington's disease. That's a little bit of a mouthful. It's Generation HD1, for sure. Now this is a phase 3 study to definitively confirm whether or not this RG6042 is effective in treating HD. So the objective is to evaluate the efficacy and safety of intrathecally administered RG6042 in adult patients with manifest HD. This is going to be a big study, if not the biggest conducted in HD to date, treatment study. There's going to be approximately 15 countries involved, so it's a truly global study, including Canada. And there could be up to 90 sites involved. So this is really a big study. We hope to launch this by the end of this year, so very soon, with patients enrolling in early 2019. The design is what you've heard a number of times today, a randomized, double-blind, placebo-controlled study. Now this design is critical to enable us to know definitively what the benefits and the risks of this molecule. It will have 3 arms. The first arm, 120 milligrams monthly, like in the OLE. Second arm, 120 milligrams every other month. And the third arm, very importantly, placebo. The study will last for 25 months, so just over 2 years. 25 months comes from just the scheduling around a bi-monthly or every other monthly scheduled dose after a 2-dose loading period. Patients will be randomized equally to one of these 3 arms. And what do I mean by randomized? I mean that there will be it's basically a flippable 3-sided coin. So there will be a randomization, a flip of coin, into one of these 3 arms. And double-blind, as we've heard in a number of sessions today, this means that your investigator, a patient's investigator, and the patient, and actually also the sponsor, so it's actually officially triple-blind. There isn't like a code kept somewhere. A vendor keeps a code somewhere, but we do not have access to that even at Roche easily. So no one knows what dose an individual is getting in the study. And this is really important to maintain the integrity of the study so we can get to the answers that we need at the end. 660 patients will be enrolled in this study. That's 220 per arm. This number is calculated based on what we call the primary outcome measure for the study. And the primary outcome measure is how we measure the success of the trial and how regulators, health authorities, Health Canada, the FDA in the US, measure whether or not this drug is effective. I haven't got it on the slide, but we may have learned that we have 2 primary endpoints for this study. We don't have 2, but we have different ones in the US versus the rest of the world. So the CUHDRS, which is this composite of the UHDRS, will be the primary outcome measure for most of the world for this study. This is a multi-domain measure, so it looks at, it measures cognition, it measures motor symptoms as well as function, and sums it up into a total score. So this, we believe, is very important for the potential of this molecule as a potential disease modifier, or a potential drug to slow or stop disease progression. You know better than I do what HD is. It's not a single symptom. It's a multi-domain disease. And it's important to be able to measure this with an outcome measure that touches these different aspects. The FDA has asked us to use a different outcome measure, primary outcome measure, and you're probably familiar with this. It's the total functional capacity, or the TFC. Now this is also an important outcome measure. The FDA's philosophy is that they want us to measure something that's directly measuring patients' function on a day-to-day basis. So some of the outcome measures in the UHDRS measure aspects that are important, but it's not exactly, is a patient still able to drive, or is a patient still able to prepare their own meals? And that's a direct measure, that's a direct function that we do every day. So what this means is that to enable us to test whether or not we have a benefit on the TFC, we need 220 patients per arm. Okay, so lastly, I just want to talk a bit about the key inclusion criteria for this study. I know this is a very high interest to you all. So the study will include clinically diagnosed manifest HD, patients aged 25 to 65 years of age, CAP score, which is a CAG age product of greater than 400, independent scale greater than or equal to 70, and the ability to walk and talk, therefore being able to participate and provide responses to the measures in the clinical trial. I'm going to take it a little bit more in detail to some of these aspects because some of them are quite new to clinical trials in HD. Before I do this, I just want a little word on the specific stage of disease that has been selected for this study. We know that HD is a continuum. We know very well that we definitely understand that as well as the adult motor onset stage, there's a juvenile HD onset, a juvenile HD with onset under 20 years of age, as well as elderly onset, so greater than 65. But for this study, we have chosen and selected based on all the information available to go ahead with early to moderate manifest adult onset. And there's three key reasons for this, and Blair and Jeff and others have already gone over these a little bit in Q questions on previous sessions. Firstly, this is a single phase 3 study. It's a very important phase 3 study. It's important that we design and get this right to answer the questions that are in front of us. We don't want to make a mistake. And we know there is a track record in doing HD studies in this population of early to moderate manifest HD. The second important point is about measurement of progression in this group. We know that in early to moderate manifest adult onset, we can reliably measure the changes in progression. We've got a wealth of data in the databases and registries, including Enroll, and so we can predict with a certain level of certainty what the changes over time will be over a 2-year study. And this is important so that we make sure that what we call power the study, that is how many patients are in each arm. So we need to have a certain level of confidence in that so that we do set the study up and design it right from the beginning. The third important point is we also want to have the potential for individuals in this study to improve, well not improve, but to show a slowing or stopping of progression. And so this subset of the HD community, we believe, has a plausible chance of seeing benefit to a molecule like RG602. Actually, just one point I do want to emphasise though is that we clearly understand the unmet medical need in juvenile onset, in elderly onset, and in the pre-manifest phase. The interest of finding a way to prevent the onset of HD at all. And these are very important questions that we are talking about and we're getting advice on and thinking about what we will do. But the most important thing is that we get this first trial right and we get answers for this first patient subset. And we'll be able to learn as we go. We don't need to wait until the end, and learn as the program progresses and start planning the next next studies and next explorations, next experiments in these other groups. So I just want to make sure that you understand that, because I think it's an important message that we're not just focusing. We're focused primarily at this point in time in this patient population for the phase 3, but we understand the need in the others, and we will be working there too. A little bit about the specifics of the inclusion criteria. I think Jeff mentioned this a little bit in the Q&A. I just want to take you through what is this CAP score. So we have a CAP score threshold of greater than 400, not equal to, greater than. We have to set the number has to be set somewhere. And what this is a measure of the length of time an individual has lived with their mutation burden, his or her mutation burden. And it's calculated by the age at the start of the screening, in the screening visit, multiplied by the CAG repeat, as well as a constant, a mathematical constant. But for the moment, I'll just focus on its age multiplied by CAG repeat. I've got some examples of how this is calculated, because I know there's interest in understanding this. The first example is a 50-year-old person has a 45 CAG repeat. So the calculation is 50 times 45 less 33.66. And that's the CAP score. So in this case, obviously, you do it, if you're not doing it on a calculator, but if you do it individually, you need to do this first step here, but it's 50 times 11.34, it's 567. Another example, so a younger individual with a higher CAG repeat, the calculation is 30 times 16.34, leads them to 490. So they mentioned this, or it has been mentioned, I don't remember if it's Jeff or Blair, but this is a really important measure. We want to, this phase 3 study is looking to show a difference in treatment benefit between the treatment arms, so 120 milligrams monthly or 120 milligrams bi-monthly compared to placebo. This placebo arm is such an important arm in this comparison. We need to be sure that the patients in the placebo arm are going to progress at a certain rate over the course of the 2 years. If they stay flat and don't change much over the 2 years, we will not be able to discern a difference between placebo and the treatment arms. So what this CAG threshold, CAP threshold does, based on the wealth of data collected in observational studies, is gives us confidence that we're enriching the study with individuals that are going to decline at this certain reliable rate over the 2-year period. The other new inclusion criteria for a clinical trial in HD that we've worked with our strategic advisors on including is an independent scale threshold. Now, as you're probably becoming aware, this clinical study is going to be complex. It's over 2 years commitment. There's a potential for placebo, receiving placebo. There's monthly lumbar puncture procedures. There needs to be a level of confidence that those individuals that are selected in this, selected and enrolled into this study are going to be able to stay in the study for as long as possible and be able to handle the complex visits and the burden, and also have the support network around them and caregivers to help get to the visit, continue on for the 2-year duration. And one way to do this is enrolling individuals that have a certain level of independence still. So we don't actually have a traditional TFC threshold cutoff that you may be familiar with in other programs. 2 minutes left. Is that 2? I need to wave. But what is an independent scale of 70 or more, and it means that individuals will have still maintained enough self-care to bath, conduct simple or limited household duties, for example. So this is a sign that they could potentially be part of the study and stay in the study for the 2 years. Okay, so the last slide is really just to highlight some of the key challenges of this study. I mean, it's a very exciting time, and we're so proud to be leading this together with you, this phase 3 study. But it's not going to be easy. It's very complex, and there's obviously a lot we don't want to get it wrong. And some of these complexities that we'd like to share is that I've sort of touched on this already, but it's really important that patients who are in this trial stay in the trial for the full 2 years. It's a long time, but we need that 2-year data to support the assessment of the risk-benefit or the effectiveness of this drug. When we, if all going well, we're able to submit to regulatory health authorities. Route of administration makes this quite complex. So monthly intrathecal injections requires a big commitment to over 2 years. So far, in the phase 1/2A and the open label extension study, generally these procedures are going very well, 20 minutes or so, and individuals are per protocol expected to walk afterwards as soon as they feel up to it to help with the distribution of the drug. And so far it's gone very well, and then the patients are able to go home not long after. But it's still going to be a challenge. This has not been done before for 2 years. And so we're working with expert lumbar puncture colleagues, Blair and Ed, to sort of build training materials to really help support the investigators that will be doing this or that administering the drug. But this is going to be a big commitment for patients as well. And lastly, most definitely not importantly, not least important, is expectancy bias or placebo effect. We heard from Linda today about this, but it's a really exciting time, as mentioned. There's a lot of hope resting on this first study of a Huntingtin-Lowering therapy. It's really important that the individuals in this study tell their physicians and their doctors how they feel truthfully. We need to know whether they're feeling the same, better or worse during the study. And it can be the case that if you are expecting, you have so much high hopes that this is going to work, that you don't want to do that. But it's important that we test this comprehensively in this phase 3 study. And that's a key reason why we have a placebo arm in this study, but even with that, it's still important that objectivity is maintained as best as possible, not only for the patients and participants, but also the investigators. I'd like to end now. With those sort of challenges in mind, it's still an incredibly exciting time. The Roche team and all the collaborators we're working with are very proud to be moving this program forward to the next stage of testing to get the answers that we're all seeking. We have many other colleagues in the field who are working on other similar or different approaches. So it truly is an exciting time for HD research and development. But we still have a number of questions to answer, and these include conducting well-controlled clinical trials to show and prove that this hopefully are beneficial and safe. So far for RG6042, it's looking good, safe and well tolerated, but we now need this global development program to take the next step in a larger group of patients across the world to see whether or not it shows benefit and hopefully slows or stops and a good ratio of dementia. So I just want to end, so that was my conclusion, so that was my conclusions of my conclusions. But what's next? And clearly this is of key interest to you all. We are frantically looking to try to get this study up and running, and the natural history study up and running by the end of this year. And so that we would start dosing in early 2019. We have shared the countries, we've shared the number of countries and sites, but the study site or country information, aside from confirming Canada and obviously the US, will be shared on an ongoing basis. And so we don't want to share, we don't want to share site names or information until sites have have a certain number of approvals in place and the infrastructure ready to start the study and to participate in the study. So please just stay tuned for this detailed information. It will be coming on a progressive basis in the coming weeks and months. Your HD specialists and the patient advocacy groups and HSC will be kept updated as we have this information confirmed. It's important to also, when considering the interest of your loved ones in this study, to talk with your HD specialists. Clinicaltrials.gov is a website that can be checked also to find that information. So with that, I'm over time, but thank you for listening. Very pleased to be here, as I've mentioned many times. It's a real privilege and honour. And you can ask Blair to help questions.
So I think we're going to ask people to go to the mic, or our mics figure out how do we want to do this. Go ahead, scream them out if you want. I don't know.
So I have a question about the first study. I understand it was a small study, you were looking at safety, it was a short duration. But based on the work that you've done on the animal models, were you surprised that you didn't see any group differences in symptoms? Is that something you might have expected or hoped for?
Not at all. The individual dose groups were very small, and there certainly wasn't sufficient power in those small groups to see change in the period of time. And if you look at an individual group, there were often only one placebo, so you're comparing one. You can't even do statistics on most of those small groups. That's actually why putting them all together as a single group allowed us to look at these things. And we were actually really surprised that there was any signal there at all. But for what it's worth, and it is just, looking at a post hoc, what we call a post hoc analysis. That means the study's done and we're just trying to learn from the study. When you look at everyone in the study, you say, what happened to the people that had the most Huntingtin-Lowering versus the people that had the least? There was a correlation with better scores. So that was a surprise. We didn't expect to see that. In fact, I wouldn't have expected to see any difference in this short period of time using the measures that we're talking about. Don't be shy. Yeah?
Quick question. The example you showed, that score. Sure. In the example you showed for TAP score, how was the constant determined? Or is that too involved?
I could tell you, there have been a number of these calculations in the past, and the CAP score specifically was calculated based on information from the PREDICT study, and it basically is just a way of, when you look at all the data, of determining the cumulative burden of disease, so you sort of can normalize people. We know that if somebody has 55 repeats and they're 45-year-old, and then 45 repeats and they're 45, they're different. And so when you look at large groups of data, you need some way to kind of calculate a constancy. And that number is just purely calculated. It's just what fit best with the data. So it doesn't have any meaning other than it's just the number that gave the best fit for the data. And there are a couple of other formulas that could be used that are slightly different, but it's just a way of determining, of sort of normalizing how do we deal with all these people with different CAG repeat sizes and different ages and different time. And it is purely generated from data. It's not.
I can calculate it to see if my wife qualifies.
Well, that constant, that same 33.11, whatever the number was, is the same for everyone. So that formula is constant. You can plug in your age and your CAG size. But there are many aspects that go into making an individual eligible. So to say that's one exclusion, the inclusion criteria.
Yeah.
So just a quick question. I was curious about, we know that the healthy Huntingtin protein has important functions in the cell. And I'm just curious if you guys saw any observational negative effects from doing total Huntingtin lowering, because that's been something that's been discussed in the past in your research so far.
So as you know, our group has done a lot of that research, and we've looked at it in many preclinical models. And there's no question that Huntingtin plays an important role in development, specifically in the development of the central nervous system. And many of the models that have raised questions about this delete or eliminate Huntingtin throughout development. More recent studies have looked specifically at what happens when you only take away Huntingtin in the adult brain. And there's some conflict in the data. Some studies have shown that there's no effect in the brain. Others have shown there's effect in the brain that doesn't replicate Huntington's. But in general, I think there's consistency that no one wants to eliminate Huntingtin at any time completely. That deleting wild-type Huntingtin is very bad in the embryo and in neuronal development. Once the brain is developed, it's unclear, really, whether or not lowering Huntingtin has any adverse effects. Now, there's no question that if you had two drugs completely equivalent that could treat everyone and were both as effective and safe, we would go with an allele-specific version, no question, if that was the situation. I can tell you that because much of this work was done at UBC with Ionis and actually worked in Dr. Hayden's lab, looked at a number of allele-specific and non-allele-specific. And as I said, when compared head-to-head, there's evidence that the total Huntingtin lowering was as effective, and there was not an equivalently specific, selective, and potent ASO that was allele-specific. So there was a very conscious decision to move forward with total Huntingtin lowering, knowing that this is a drug that can be stopped, it's reversible, and it's titratable, and we're not going to eliminate Huntingtin completely. Another thing that's important to understand is when we talk about Huntingtin, the mutant Huntingtin has this toxicity, but it is still Huntingtin, and it still works as Huntingtin. So we've replaced all of the normal Huntingtin with mutant Huntingtin, and none of those developmental abnormalities occur. You can survive with just mutant Huntingtin. There are people that have 2 copies of mutant Huntingtin, and they survive throughout development. So mutant Huntingtin is not a loss of function. So when we decrease total Huntingtin by 50%, you still have 50% functional Huntingtin, even if half of it's mutant. That's important to think about.
There's a question via text. Question for you, Lauren. Is there plans to, or thoughts to expand the inclusion criteria?
For this particular study, I would assume. So no, this is the inclusion criteria as I've outlined for the phase 3 study, for this particular phase 3 study. So any sort of changes to this I don't see any sort of ways in which they would be changed. We've worked a lot with our advisors, with Blair, the Strategic Advisory Committee, and delving into the previous data that's been collected, and this is the inclusion criteria that we believe will set this study up for the best chance to test the hypothesis and for the benefit and safety of this molecule.
I would follow up, and you know, everyone has to understand that this is not the end. This is not the last study. What we're not, you know, we're really doing this trial to answer a question. There is a lot of discussion about having perhaps even parallel groups that may start later of other groups that might be of interest. So as a possibility, I'm not saying this will happen, you could have a parallel group with a pre-manifest individual that are followed. So this is the start of the process. One study isn't the end. We're going to continue to work and try and ensure that this is that the best therapy gets to all HD patients.
And I'll just add on that, thanks Blair, that clearly this is the inclusion criteria for the phase 3 study. And as mentioned, we are looking into parallel activities. But if all goes well and the hope pans out that we do see that this drug does lead to clinical benefit in patients, won't be limited to those the very constrained criteria within the phase 3 study. There may be limits. It depends on what the health authorities would approve. Like they look at the criteria, they look at who've been studied, they look at how generalizable the findings will be to the broader HD population. So there would be a discussion about that, but it most definitely would not be limited to the inclusion-exclusion criteria from the phase 3 study. This is a study that needs to have some constraints and limits to allow it to take place and to be executed. Yeah.
Mark?
So speaking as a patient advocate and board member, Lauren, this is for you. You're probably aware that the Huntington side of Canada has spent the last 7 or 8 years with their clinical trial readiness program, creating maps on our website where sites are. And I think Roche is working hard to try and cover the country as best as possible. So the question comes in, you know, there's people coming from all sorts of areas. What is the Roche travel policy for people coming into these studies? What kind of limitations will there be as far as having travel reimbursed and paid for? So it's not just the immediate geographic areas around the sites that people can participate.
It's a very, very important question. Now, Roche does not put any limits on geographical scope for an individual investigator or an individual HD specialist. An individual clinic or centre will have their own policy of who they would want or look to have in such a clinical trial. That said, there are some limitations, and we as, I must say I don't know the specifics, but we do have support programs that are in place at each individual site is able to use, and then individual patients will have to cover reimbursement within country, and there's local laws that dictate how much is able to be reimbursed for a clinical study like this. So there are some boundaries, but again it will be a very important discussion with the HD centre and specialists as to what the scope and breadth of the reach. Did you want to add something?
No, it's very important for people to know up front that it's not just the immediate area, and as far as the information that I have, it's going to be based on individual aspects and total costs and things like that. Thanks.
And there's big recommend
I was curious about the response, the individual response, that some of course there are variations in how much the dose get, and some of them didn't seem to respond at all. And so I'm wondering that when you have this tiny piece of DNA, I assume that you were targeting the common I guess, the shared sequence between the different individuals. I guess, so like, where do you get that data? And I'm just wondering that it also applies to any ethnicity or any different genetic background people.
Yeah, so excellent question, very insightful. The sequence that is being targeted, as far as I know, does not contain any known SNPs. So it should not vary between ethnicities or between populations. It should be generalizable. And in fact, there was one individual at the highest dose that looked like it went up. But if you actually look carefully, what you'll see is it went up and then it went down. And in fact, it had a very, very low initial value. And it's probably that initial value that might have been incorrect, because there was a response and that person's been followed out, and in fact, there was lowering. We did actually very carefully look at that individual and sequence them to determine whether or not the region that the ASO binds to had an alteration, but it didn't. So, wonderful question, but no, this should be applicable across populations, and as far as I know, and I can't state with 100% certainty, there is no known variance in this binding region.
Were people that were participated in the first study eligible for the next one?
So all of them are actually enrolled in an open label study already, and so they're receiving treatment. So they are not eligible, but they don't need to because they're getting treatment already.
And all 46 individuals who participated are continuing on active drug now, and they're all, there haven't been any dropouts.
We're getting this signal.
Just like you guys. All right, well, thank you.
Well, that was really, really incredible. I'm sure you enjoyed it and learned as much as I did. We're so grateful in the HD community. We have such brilliant scientists and dedicated, compassionate doctors. We met two of them today. Blair, who's been a longtime friend. Thank you so very much.
Thank you for the opportunity. Really appreciate it.
Warren, who did such a phenomenal job of making something very complicated understandable. Thank you.